All right, if you're watching this, that means obviously you're watching the video. No one's here in the room yet, and no one's live streaming. So if you're watching the video, it's going to be business as usual. I'm going to give you a series of attendance words and phrases. Every time I do, you need to write it down, take a screenshot, and then at the end of the video, you're going to, or once you're done watching the video, you will send me the list of um, attendance words with the accompanying screenshots. The last word for attendance is going to be me asking you to ask a question about the material covered in today's lecture, as usual. So that being said, today's first word for attendance for you is going to be plug. Like you would plug something in, or somebody might get a hair plug. But yeah, that's it. And it is currently 7:25, so you can skip ahead about 35 minutes, because that's when the rest of the lecture will start. Who's the football team playing this weekend?
Martin College. Oh,
Let's see. Brumfield is here. Wait, no, not right. Champion is here. Gorby. Gross. Gunther is here. I sworn Rainbow was here. Bill is here. Jackson. Not here. Keys. Not here. Perez is not here. Sigmund. Nope, not here. Slaughter. Not here. Smith. Not here. Stover is here. Tompkins is here. The Nest is not here yet. Watson is here. Young is here. Zweibeck is here. Lawrence is here. Carter's not here yet. Smith just came in, correct? Right. Got less than 2 minutes. Let's see who else shows up. Other than football, does anybody else have plans this weekend? Anything worth mentioning? Hopefully this lecture will be just slightly quicker than the rest. Maybe like 5 minutes or something, we'll see. Morning. Keys, right?
Yeah.
Okay. And Sigmund, correct? Okay. And that's 8 o'clock. It's not 8:01 yet, though, so no one's technically late yet. So as always, I'll remind you guys, of course, I'm preaching to the choir. Attendance is mandatory and is graded. It's going to be an entire letter grade. You know, it's 100 points out of 1,000. So you guys who are usually here are sitting great. You guys know people who, you know someone in this course who hasn't been coming, you might remind them that, you know, it will hurt their grade. That it was meant to boost your grade, not hurt it. So just remind them of that if you know them. And of course, as usual, I give a bunch of attendance words. If you're here, you don't have to send them. But if you do send them, you can get extra credit. I'll have to have those words by 9 a.m. to get the extra credit. I mean, otherwise, you could just, somebody could just tell you what they were. I was asleep. What were the words? And then for those of you who are watching the video, no one's with us live right now, but if you're watching the video at a later date, as always, you're going to want to take a screenshot when I say them. And then at the end, it's going to be one question where I say, hey, ask a question about the material in this video. So yeah, business as usual as far as that's concerned. So the first word for attendance. Someone give me the first word for attendance. What you're trying to think.
Only.
What is it?
Only.
Only? Okay. Only is the first word for attendance.
That was just the first one.
I like it. Only. Don't think I've ever used it before. So yeah, there you go. Only is the first word for attendance. All right. Anyway, moving forward, to remind you, my office hours are virtual only today because I'm traveling. I'm leaving here and getting on the road. So,
Where you
Pittsburgh for a wedding.
going? That's only about 2 hours.
I think it's 4 last I checked. The way my wife drives, it might be 2 hours. Anyway, it's gonna be, I'll be on the road. So, you know, try to reach me online, or call my office, which is forwarded to my cell phone. Again, because I'm traveling, I might be out of service. So if you can't reach me, I apologize, but I'll try to get back to you. So any questions about that? All right, don't forget the pre-lab 3 is due today. Make sure you get that in. Better to get it in incomplete or horrible than not get it in at all, right? Because you can always make corrections. This one's a little bit different. One part that's not different is that you have to do a video first, so make sure you do the video first and then do the rest of the lab. Then the part that makes it different is now you're gonna go ahead, like once you're done with the pre-lab section, you just keep going a little bit and you're gonna answer some hypothesis questions. Because the next week's lab, you're gonna look at
all these different materials, like starch solution, glucose solution, fruit juice, things like that. You're gonna say, all right, does it have any monosaccharides in it? Does it have any polysaccharides in it? Does it have any proteins in it? Does it have any fats in it? So today, if you haven't done it already, that's what you're gonna do is put down your hypotheses. So any questions about that? All right, but for any of this, don't forget I'm here to help. I was just thinking about that because of an email I got yesterday that reminded me of some stuff I saw from student evaluations from last semester. And it kills me when someone's like, it was too hard, I didn't get it, which I understand that. But if it's too hard and you don't get it and you never come to me, I don't think you're allowed to say it's too hard, I didn't get it. Like, let me help you. So I'm here to help. I'll meet you online. We can go step by step with these pre-labs. All right, read this, let me know what you've done. All right, good. Now, I'll answer these questions. Let me know if you have any questions, right? I can just walk you through it. I'm happy to help. Because I want to teach you. I'm not trying to test you. I want to teach you. So you're doing me a favor if you allow me to help you. So basically, I'm repeating myself. Anybody have any announcements, reminders, questions about my announcements or reminders? All right, let's get back into it then. Let's pick up where we left off. I told you I wanted to explain this a little bit more because this is a very commonly asked question on the exam. I'm gonna give you a question about pH. Maybe I'll give you this picture to refer to. So I might say something like, how much more acidic is, I don't know, pure water than milk of magnesia? It might be something like that, because this one's out of whack. So I'll either give you the picture, or I will give you the numbers and say, how much more acidic is pure water, and then like parentheses, pH 7, than milk of magnesia, parentheses, pH 10. But the thing you need to know, remember, here is every step is a multiple of 10. So if we're going from 7 to 8, that's 10, right? That's 10 times more basic. 7 to 9, 9 is 100 times more basic than 7. 7 to 10, 10 is 1,000 times more basic. So every time you move a step, you have to multiply it by 10. And the part that gets tricky, and I'm not trying to be tricky, I just want you to think and not memorize, is you have to think about all the different ways I could ask that, right? I could ask, how much more acidic is pure water than milk of magnesia? Or I could ask you how much more basic is milk of magnesia than pure water, right? So I could go either way. And then I might say how many more, I could talk about the H plus ions, because remember, the lower the pH, the more acidic it is, the more H plus ions it has. Or I could say, how many OH minus ions, because remember, the more basic it is, the more OH minus ions it has. So keep that in mind. I could ask one question many different ways. So just, try to be aware of that. But if it's overwhelming to you, also keep in mind there's going to be maybe one question like that. Maybe two, probably just one. So if it's overwhelming, just think to yourself, who cares? This is one point. And also you'll get to redo your tests, right? As always, once the exam is done, you have 50 minutes to take the exam. But then after that, you can retake it as many times as you want. You can use your notes. It's untimed, so shouldn't be any pressure. Anyway, any questions about that? All right, here's a meme I found, and I love it because I know it's an exaggeration. It's actually meant to be a joke, but it is, people do try to scare you like this. They'll try to scare you by telling you the truth in a scary way, right? So dihydrogen monoxide is an acid with a pH level of 7. I mean, it's in the middle, so technically you could refer to it as an acid or a base. It's all relative. Dihydrogen monoxide, sorry, I should have started there. That's just a different way of saying water, right? So anyway, dihydrogen monoxide is an acid with a pH level of 7. That's a higher pH level than any other acid, right? All that's true, but the way it's being depicted is like trying to make you scary. But think about it. An acid, the lower the number is, the more acidic it is. So the fact that it has the highest pH levels actually means it's not scary. But even though it's just a joke, this is how people try to scare you. They'll use true statements to try to make something seem scary. Anyway, obviously I'm not going to test you on memes. Wish I could. Let's talk about stuff you should need to know. I wish I could have done this. We were so close because this is where we left off on Wednesday. And we were almost there. I wish I could have taught you this before we did lab. But buffers, these are substances that minimize changes of pH. So you need to know that for the exam. How do they do it? They basically just do the opposite of what's happening. So we know that an acid is something that gives out H plus ions, right? So if you put an acid in it, it's going to accept those H plus ions. The opposite is true when you put a base in it, right? So a base, it's going to donate these H plus ions. And that's all you need to know. And really quickly, because I didn't get to talk about it during lab on Wednesday, this is important because without buffers, you would die. You know, going back to this very quickly. Human blood, it's not showing the number, but the pH of human blood is 7.4. If it goes a little bit above that or a little bit below that, you'll die. And for extra credit, if you want to look that up and tell me how much is too much, how much is too little, you can do that for extra credit. But anyway, you'll die if it gets too high or too little. Definitely 5 would kill you, right? So think about though, I'm drinking coffee right now, and I'm not dead yet, right? Tomato juice, grapefruit juice, soft drinks, these are all things, lemon juice, these are all things that people drink all the time, but it doesn't kill you. The reason is because you have buffers in your body, right? So it protects your blood from changing pH too much, which is another reason why people who try to sell you crap, like it's alkaline and it'll adjust your pH in your body and you should drink this and pay extra money for this water. It's BS because your body takes care of itself, right? Don't let people trick you. Anyway, moving forward. Here we go. This is the last portion. I hope we can get through it today. I'm sure you guys can read that, right? That's 2.3. So the third section of this chapter is biological molecules. This is perfect timing because this is what the pre-lab is on. This is what next week's lab is on. So I'm going to kind of go through it quickly just because I want to get the information out there so you'll be prepared for the lab. But if it's too quickly, you can meet me sometime afterward and I can re-explain any of these concepts. I really need to fix this. All right. So, but the objectives for this portion are, by the end of it, you should be able to describe the ways in which carbon is critical to life. So you already started a little bit, well, not the critical to life part, but you've already been very well introduced to carbon because of this lab we did this week. You should be able to explain the impacts of slight changes in amino acids on organisms. That'll be new to us. We haven't talked about that yet. You should be able to describe the 4 major types of biological molecules, and by the end of next week's lab, you'll have them memorized, I'm sure. And you should be able to understand the functions of the 4 major types of molecules. So here we go. Here's some things you need to know. I already told you this before, but cells are mostly water. You already know that. As a matter of fact, specifically, I told you earlier the number that you need to know. They are 70 to 95% water. So if you're taking notes, I wouldn't even write that one down because that's not specific enough, right? 70 to 95% water to be specific. So that bullet point is more of a reminder. So then, if it's mostly water, what's the rest of your cells made up of? And the rest of it is made up of carbon-based molecules, mostly. So a lot of what we're going to talk about is based on some carbon-based molecules. Not that you need to know this, but this is just an introduction. The carbon forms a lot of complex molecules. You already know when we were talking about the big 4, right? Hydrogen bonds once, oxygen bonds twice, nitrogen bonds 3 times, and carbon bonds 4 times. So when you think about it, if you're building something and you can stick things to something, obviously carbon is going to be more complex, right? Because carbon, you can put 4 things into it. So imagine all the different possibilities as compared to nitrogen or oxygen or hydrogen. So again, this won't be a test question. This is more of an introduction. Like think about why is it that carbon, why is everything carbon-based? And that's what we're getting at. Anyway, organic compounds are also known as macromolecules. So you're going to learn a lot about macromolecules. I'm almost definitely not going to ask you the definition of it. Actually, no, I'm not. But yeah, we're talking about macromolecules for the most part in this portion of the chapter, which basically just means big molecules. So most of the ones you drew for the lab were not macromolecules. They were carbon-based, mostly. But they weren't all macromolecules. Anyway, again, let's talk about carbon. It's a versatile molecule. Again, as you already know and need to know for the exam, it bonds 4 times, right? It makes 4 covalent bonds, whether that's 4 single bonds or 2 double bonds or a triple bond and a single bond, whatever it is, right? It bonds 4 times. Because of that, it can make this endless diversity of carbon skeletons. And these carbon skeletons vary in their size, their bond branching pattern, all kinds of stuff. There's nothing to write down here. Sorry, I should have told you that before I started. This slide is still just like an introductory slide. Like, hey, what are we getting into? Of course, again, you do need to know that bullet point. carbon does bond 4 times. But again, just an introduction. Here's an example from your old textbook. These are all pretty small molecules. They get really large, and these are just a few 4 examples. So here's a nice simple carbon molecule that only has 2 carbons and some hydrogens. This one's got also 4. This one has 4 carbons. Some of them go horizontally, some of them go vertically. This one's in a ring. It has a bunch of double bonds and single bonds. This one also has some double bonds and single bonds. How many of you are chemists? I think I've already asked this before. None of you are chemistry majors, right? None of you are chemists. But what you can tell by this, and this will be a theme in this course, even though you guys aren't chemistry majors and you probably don't understand chemistry that well, what you can see just by common sense is, look, these things have different shapes, right? And a lot of what happens in biology is this molecule fits in with this molecule in this certain way. So it's a lot like Legos or something that building blocks. So just by looking at this, you can tell that these things must do different things, right? They have different shapes. They fit into things differently. So that's another thing. There's a lot of structure and that we'll be talking about, especially in this portion of the semester. But then throughout later in the semester too. Structure and function, right? So we see these different structures, and you can tell that they have different functions. As a matter of fact, you could probably guess something like this and this. They probably have similar functions, but slightly different, right? Because they have similar structures, but slightly different. But this compared to this, I bet this thing has a completely different function than this. And it's just common sense. And of course, you're not going to be tested on that. But when you see things like this in this part of the semester, just keep that in the back of your mind. Just look at the shape and just kind of use common sense. Even though you're not a chemist, just think it's completely different shapes. So they must do completely different things. Any questions about this? All right. So again, this slide is just an example. I'm not going to ask you any questions about those particular molecules. As you already know, carbon can bond with other elements, most commonly hydrogen, oxygen, and nitrogen. You're now very familiar with that from the lab. Again, that's the big 4, right? You need to know the big 4 for the exam. Oops, excuse me. You need to know the big 4. Excuse me. Yep, okay. Yeah, anyway, there's a little star asterisk here. Not that you guys need to know, but just so you know, carbon also bonds with a lot of other things. But when we're talking about biology and organic chemistry, this is the 4 things it bonds to. So as far as we're concerned in this class, those are the things that it most commonly bonds to. But just so you know, it's not always the case when you start looking at other types of chemistry. Here's one example. You don't need to write anything down about this. This is just an example. But methane is like one of the simplest organic compounds because it's just that 1 carbon, right? And carbon bonds 4 times. So it's just 1 carbon bonded to 4 hydrogen atoms. Nice and simple, right? Not that you need to know this, but just since it's an example, we're giving you a little bit more information here. It's abundant in natural gas. It's produced by prokaryotes. And I don't think I've introduced you to that term yet. Yeah, I have. But anyway, to remind you, prokaryotes are bacteria, right? So these swamp bacteria, they produce swamp gas, and it's methane. You can also find it in digestive tracts of grazing animals, right? So cows, this is why their farts are, put out so much methane. Actually, not just that, too. You can look into humans. Sometimes humans produce methane when they do that. Anyway, any questions about this? Just an example. All right, here's different ways of looking at it. You should be familiar with this, right? This is the way you've been drawing in lab. There's different ways. It's the ball and stick model, space filling model. You do not need to be aware of those. I'm just showing you that, again, that there's different ways of drawing molecules. So larger organic compounds are also the main molecules in gasoline. I'm not going to ask you that, but it's an interesting comparison that either your textbook or the old textbook, I don't remember which one, but they make this comparison, and it's a good comparison. Because not only is it the main compound in gasoline, but it's also the same stuff that fuels your body and your cells, right? Because there's energy-rich parts of the fat molecule that have a structure similar to gasoline. And here we go again, like I keep saying, with structure and function. They have a similar structure, at least at this level, in 100 level class. You can guess they might have a similar function. Not always, but it doesn't hurt to make that guess. But anyway, here's what octane looks like. That's what that molecule looks like. And remember, I was saying something earlier last week. The more energy it takes to build something, the more energy is released when you break it. So basically, and this isn't always the case, but the bigger something is, right, like this long octane molecule, it's going to take more energy to build it because it's just bigger. Therefore, it's going to take release a lot of energy when you break it. So that's octane. Here's dietary fat, and you're going to become familiar with this in this portion of the chapter. So dietary fat almost kind of looks like 3 little octanes stuck together. And in a way, in a very basic way, it is. And again, once you break down those molecules, it releases energy. Anyway, any questions about this? All right, the next slide, I'm going to show you what the actual drawings of these molecules look like, and you can see how similar they are. So here's octane, right? And this is one of the saturated fat. So that's slightly different than the picture I showed you. The picture I showed you was 3 of these things stuck together. But nonetheless, you can see how they're very similar, right? And again, similar structure, similar function. Big molecules squished together, right? When you build these big molecules, it takes energy. When you break them down, it releases that energy. All right, so we're still talking about carbon chemistry. I'm not going to test you on this. This isn't in your book, but it's good information because it helps you later in the semester. If you keep this stuff in the back of your mind later in the semester, it will help. The unique properties of these carbon molecules depends a lot also on the carbon skeleton, partially, but then also to the things attached to the skeleton. And those are called functional groups. Now, I'm not going to test you on that, but I am going to use that word throughout the semester, so I need you to know what a functional group is. And again, at this 100 level class, it's just stuck. It's just stuff, other molecules that have been attached to the carbon skeleton. I mean, the official definition from the old textbook is groups of atoms directly involved in a chemical reaction that are attached to a carbon skeleton. And that's true too. But again, as far as we're concerned, you can just think, you've got this carbon skeleton, but a lot of what makes that molecule unique, makes it do what it does, is these functional groups that are attached to it. For example, this slide that you don't need to know at all, it's just an example, right? Here's a bunch of different functional groups. Hydroxyl groups, methyl, carbonyl, carboxyl, amino, phosphate. These are all different groups. You're going to be exposed to some of these, but you'll never need to memorize them. But once again, going back to what I showed you before and talked about before, even though you're not chemists, look at all these different shapes. That's a different shape, different shape, different shape, slightly different shape, right? These all have different shapes, so even though you don't know anything about them, you can imagine they do different things. One thing we haven't talked about yet, but we're about to talk about, hopefully today if we get there, is the difference between polar and non-polar as far as chemicals are concerned. What happens when you mix oil and water? Does anybody know? They separate, right? And that's because, as I'm going to teach you later, water is polar and oil is non-polar. So polar and non-polar don't mix. I only say that because if you have a molecule that has this thing on it, but it also has this thing on it, then that part of the molecule will repel itself, right? So you won't have a molecule floating up on itself. It'll repel itself at that point because these things don't go together, right? Some other example. Some of these are charged, right? So if they have a positive charge and then something else is negatively charged, they're going to be attracted to each other. If something's positively charged and the other thing is also positively charged, they're going to be repelling each other. So we're never going to get that deep into this class for the most part. But the point is, these things have different shapes. Some of them are charged or uncharged. Some of them are polar or non-polar. So all these things dictate what they do when you put them together. And when you put them together, that's what chemistry is, right? What do these molecules do when you put them together? All right, you don't need to write this down yet either. This is just an introduction to what we're about to dive into. But on a molecular scale, many biological molecules are gigantic, earning the nickname macromolecules like we talked about earlier. I've already introduced you to that term. There's 3 different categories. Again, you don't need to write them down because we're going to talk about them. There's carbohydrates, proteins, and nucleic acids. These are all macromolecules. Hopefully you're reading this and you paid so much attention to that pre-lab that you've already done that you think to yourself, wait a minute, wasn't there a 4th category? And there is, and the 4th category is lipids. But technically lipids are not macromolecules, and we'll talk about that when we get there. Any questions? All right, anybody want to tell me the next word for attendance? All right, proteins then. Proteins is the next word for attendance. All right, so what are macromolecules? Here's some terms you need to know. Maybe because I'm going to test you on the definition, but definitely because I'm going to use them. Macromolecules are called polymers. Polymers are made up of monomers. So a polymer is a big molecule, right, a macromolecule that's made up of different parts. Those different parts are called monomers. As far as we're concerned, they're basically the same parts. Monomers are like repeating parts. Like a train would be a polymer. And each train car would be a monomer, right? Attach this train car to this train car, this train car, this train car. And before I move forward too, I keep this in mind as I'm teaching the rest of this chapter. For the exam, a lot of these questions are going to be, all right, is this a carbohydrate? Is it a lipid? Is it a nucleic acid, or is it a protein? Right, you're going to need to be able to compare and contrast between the four on the exam. And one of the things you're going to need to know the difference about them is what their polymer is called and what their monomers are called. So even though you don't know it yet, well, you might, even though I haven't taught you that. If I say, all right, which of the fours has the monomer of amino acid? By the time you take the exam, you're like, oh, amino acid monomer? That's a protein. Which has a polymer of, I don't know, glucose. And you say, okay, well, that's a carbohydrate, right? So you're going to need to know that eventually. So again, as I'm teaching this, try to remember what the polymers are called and what the monomers are called. Because that's going to be on the exam. That's going to be part of how you differentiate between the four different categories for the exam. Here's another term you're going to need to know for a good portion of the semester and for the exam. You need to know there's two different types of reaction, and the first one is called a dehydration reaction. So you need to know that. A dehydration reaction is basically when you bring stuff together. Yes, it says it links two monomers together, and that's true. But the easier way, in my opinion, to remember is if you're building something, right? I keep saying building a molecule, building a molecule. If you're bringing stuff together, it's a dehydration reaction. Why is it called a dehydration reaction? Because it removes a molecule of water. I'm going to X do that. I'm not going to ask you if it does that. That's just my way of helping you remember that a dehydration reaction, it is what it is. And the next slide I'm going to show you might help you remember this too. So the next slide I'm going to show you is an illustration of a dehydration reaction. Excuse me. But before I move forward, does anybody have any questions about this slide? All right, and again, you need to remember polymers and monomers. As we move forward, you need to know, again, the polymers of these macromolecules, the monomers of these macromolecules. When I use the term dehydration reaction, you need to know what I mean by that. We're building something. So the next slide shows what I'm talking about. Here we have a short polymer attached to a monomer. It could be anything, right? It could be two monomers put together. It could be two polymers put together. The point is, as far as we're concerned in this class, whenever we're bringing molecules together, it's a dehydration reaction. And you can see here, not that you would need to know all these details, but I'm just trying to help you remember why it's called a dehydration reaction. It's this monomer happens to have a hydrogen on that end. This polymer happens to have an OH on that end. So when you put them together, those go and do their own thing, and that forms a molecule of water. So and look, when you look at the products, right, there is no water in the products here, at least not for that molecule. Right? So that's what we're dehydrating. Right? We're taking water out of the reactants. That's pretty easy to remember, right? Dehydration reaction brings things together, removes a water molecule. I always thought that was easy, but then it gets trickier because I have another word for you. See if anybody can guess it. I don't know. I would be surprised. If this is what it's called, if it's a dehydration reaction when you bring something together, what do you think it's called when we break it apart? Great. I'm so glad you said that. It's not, but it's close enough, right? So if you can remember that, right? Dehydration, when you bring it together, you remove a molecule of water. The opposite of that, this sounds a lot like hydration, but it's close, right? It's actually called hydrolysis. So if you can remember that, hopefully that'll help. And it's the opposite, right? So hydrolysis reactions, they're breaking things down, right? Dehydration reactions are building things. Hydrolysis reactions are breaking them down. Dehydration reactions produce, they take water from the reactants. Hydrolysis reactions use water, right? So they're using the water to break these molecules apart. Another way you can remember this, I don't think I have it written down, so I'm going to go ahead and write something down here. If we have I don't know, molecule A plus molecule B. Like if you didn't see any chemical reaction like this, and you might for the exam, of course, it might not be A and B. The point is, it doesn't matter what molecules. I might give you a molecule you've never seen before. Bear with me. You get molecule A plus molecule B gives you molecule AB plus water. You know that's a dehydration reaction, right? Because you have taken water, you've dehydrated the reactants, right? And another way you know is because here you had A plus B, here they're together. The opposite would be true if you had something like, you know, CD, molecule CD, it would stay on there, molecule CD, and then you have, oops, oh, come on. Hopefully you guys can picture it because this, I swear, no. Throw that computer out the window. Anyway, it's not letting me draw, but just try to imagine maybe the opposite of this then. Forget that. Imagine if I just took that arrow and flipped it. If you have hydrogen, or excuse me, water on the reactant side, right, that would be hydrolysis. So any questions about that? Okay. Making pretty good time here. You do not need to write anything down on this next slide either. Once again, I'm introducing you to the 4 things we're about to talk about. We're about to talk about carbohydrates, lipids, proteins, and nucleic acids. For the exam, you need to know the difference between the 4. So you're going to need to know their polymers. You're going to need to know their monomers. So you're going to need to know their functions. They do different things in cells, so you need to know what they do. And sometimes there'll be something specific about them that's different than the rest that you'll need to know for the exam, and I'll tell you. So, does anybody know what this is by any chance? Not that you need to, it's not important for this class.
Tardigrade.
Yeah, it's tardigrade. Sometimes they call them water bears. It's like the smallest animal in the world. They're microscopic. They're really interesting. But it's not the smallest organism, but it's the smallest animal. You might know what this is. Obviously, it looks like a bunch of trees, right? Technically, that's one big tree. So there's one grown by seeds. That's one big tree connected at the roots. My point here is whether you have something that's alive that's really, really, really tiny, or something alive that's really, really, really big. So again, that's one organism. I know it looks like a bunch of trees, but that's one organism. No matter what, this is something that all living things have in common. These 4 macromolecules. And that's why you're learning about it. So the first one we're going to talk about is carbohydrates. All right, so if you were in biology class and someone's talking about carbs or carbohydrates, what would you think of? If someone said, I'm going to go get some carbs, what do you think they're talking about? You think they're going to Hamlet Hall to get in the lab to get a vial of carbohydrates, or do you think they're saying something else? Any guesses? Yeah, something else, right? Like what? Like when in regular everyday life, someone says, I'm going to get some carbs, or I need to cut out some carbs, or something like that. What do you think? There's a lot of carbs in that. What do you think they mean? Don't be shy. Surely you guys know this. You just be too shy to talk. No one? Even athletes? You guys don't carb load? Well, I guess you do. You don't do long distance athletics. But all right. Hopefully you guys know this and you're just being too shy for some reason. Trying to get over that. I hate talking to you. I'd rather talk with you. Usually when people talk about carbs, this is the thing I'm thinking of, right? Spaghetti, right? Pasta, that's carbs. But then also, not just the spaghetti, but also that soft drink, right? The Coca-Cola. So carbohydrates include sugars and polymers of sugars. So if you were doing a flashcard for that, you can put carbohydrates on one side, and on the other side you should put sugars. Now, it does get a little bit more complicated than that, but for the exam, if you see anything about sugars, you should think carbohydrates. So again, for some examples, as you can see on this picture, we've got these small sugar molecules that are in that soft drink. And then we have these other molecules, the longer molecules that would be in spaghetti or bread, things like that. You don't need to write those examples down. We're going to talk about it here in a few. But yeah, so again, when you think of carbs, you should think of sugars. So even that starch in that pasta or bread, those are sugars, right? Rice, all that starch. Starch is just a bunch of sugars linked together. Here's something you do need to know for carbohydrates, because I'll ask what the purpose is. In animals, the purpose of carbohydrates, it's just the primary source of dietary energy. That's the most important one. But it's also the raw material for building other molecules. But for the exam, you need to know that first molecule. For animals, carbohydrates are the primary source of dietary energy. Of course, actually, there's going to be another slide where we get a little bit more specific. So now that I'm thinking about it, if you're taking notes, I guess you could not even worry about this slide yet, because we're going to get deeper into this than this slide goes. But anyway, moving forward, in plants, it's also the building material for much of the plant body. But as you're also going to learn as we move forward in this lecture, it also provides energy for plants. But for extra credit, you can look something up, because what the world isn't just filled with animals and plants, right? There's other things too. Bacteria, protists, things like that. So if you want to look up fungi, right, you could look up what do carbohydrates do for these other life forms other than plants and animals. So any questions about that? All right, let's start talking about how they're built. So the monomers of carbohydrates are monosaccharides. You need to know that for the exam. Those are the monomers. Monomers of carbohydrates are monosaccharides. And it's kind of easy to remember. Well, sort of, at least for now, until I introduce you to the rest, right? Because monosaccharides are the monomer that goes together. Saccharide basically comes from, I think, the Greek word that means sweet or sugar. So there you go. So this is as small as it gets, right? These are the monomers. You cannot break them down anymore and still be considered a sugar. Of course, you can break them down because they're made up of obviously carbons and hydrogens and oxygens. You can break them down into the parts, but if you break it down anymore, they're no longer sugars. So again, these are the monomers. They're the smallest sugars we get. They usually end with ose. So I'm not going to test you on triose or pentose or hexose, but if I ask you any question, I'm like, when I give you some word, I'm like, what is this? Right? And if it's a word you've never seen before, then it ends with ose or oses if it's plural. And I tell you, it's a sugar, then you say, okay, well, that's a carbohydrate. It's the monomer of a carbohydrate. So yeah, let me make sure that's clear too. I hope that's not confusing. I just realized how that could seem confusing. Monosaccharides are themselves carbohydrates, right? So we get a triose or a bunch of trioses. You get a triose, that is a monosaccharide. But it's also the monomer of carbohydrates, like larger carbohydrates, larger polysaccharides. So again, I'm not going to ask you about triose, pentose, or hexose. We'll make sure we're still recording. Excuse me. So far, it's actually worked today. Amazing. Anyway, some common examples, not that I'm going to ask you, but you can get glucose in sports drinks, fructose in fruit, and then you can find both of these simple sugars in honey. And what's interesting to me, like I think when most people think of sugar, and we'll get to this here in a few minutes. When most people think of sugar, they probably think of table sugar. Table sugar is actually a disaccharide. So when you're drinking, Gatorade, for example, or honey, that's actually a monosaccharide. So anyway, here's the next example, and another term you need to know. So you don't need to know this example, but you need to know this term I'm about to introduce you to. This is a picture from your old textbook, or the old textbook, so you won't have this in your textbook, but this concept is taught in the textbook. You need to know what an isomer is. An isomer is something that has the same formula, but different arrangements. And you're already familiar with that. If you guys were to look at all your different drawings from the lab, you guys had the same formula, but you guys sometimes drew them different ways. That's okay. Those are called isomers. And again, going back to this structure and function thing, you can look at glucose, you can look at fructose, and you can see, man, those things are really, really similar, but they're slightly different. And that should tell you, rightly in this case, that they have a very similar function, but slightly different. For example, fructose is sweeter than glucose. And the reason it's sweeter than glucose is just because of this little difference right here, right? So our taste buds pick up those molecules a little bit different. Those molecules fit into those receptors a little bit different. And because of that, your brain picks it up and says, oh, that is sweeter than that. Not that you need to know that for the exam, but again, it just goes to show you how a slight difference can make a big difference. A slight difference in the shape can make a big difference in the function. Anyway, any questions about that? So again, formally, here's everything I just told you, but in writing. Glucose and fructose are examples of isomers. Isomers are molecules that have the same molecular formula, but different structures. And again, this goes back to people trying to scare you. You know, people trying to say, well, you know, that's the same stuff they put in this, blah, blah, blah. You don't want to, I can't believe you're going to put that in your body. It's poison. We should ban it. Stuff like that. Because maybe they have the same chemical formula, but they could be completely different things. Some of you might be familiar with the example I'm going to give you on the next slide. I'm not going to ask you any questions about it for the exam. It's not on your textbook. Actually, I remember seeing it on a television show and thought, hey, that's cool. I talk about that too. But anyway, I know this is a really complicated looking molecule, but here's two different molecules. They have the same chemical formula, right? They look the same. They're actually just mirror images of each other. One's called L-methamphetamine, the other one's called D-methamphetamine, right? So they're the same thing, I mean, the same chemical formula, and they're only slightly different. The only way they're different is that they're mirror images of each other. Yet they're very different in what they do. The one on the left is used in things like Vicks VapoRub, right? Anyone want to guess what the one on the right is, based on the name? Any guesses? Man, you guys are too shy. Surely somebody can guess this too. D-methamphetamine. No guesses? Meth? I hope someone said meth. Good, yes. Of course, that's just Breaking Bad. But yeah, so one's used in medicine, the other one's used for people to get high, right? There's a big difference, huge difference in what they do and what they do to your body, but it's the same chemical formula. Again, it's the same, even the same structure, just mirror image of each other. And this is a really big deal, not for the exam, but for everyday life, because again, people will try to scare you with stuff like this, and well, in this case, it is a big difference, but you know, imagine someone saying, like, no, I can't believe you're gonna rub that on your kid's chest. That's the same stuff that's made of meth, or the meth is made of, and I guess you could argue that's true, but now that you know, yeah, it might have the same chemical formula and the same shape, but it's not the same. I see crap like that all the time. Like one of them was, what was it? I think there's a medicine out there called Miralax, polyethylene glycol. They're like, oh, that's only one molecule away from being antifreeze. Like, okay, well, that's one molecule away, so that's a huge difference. This is just the same molecule, just a different shape. So yeah, having that one molecule, it's one molecule away. Yeah, that doesn't scare me, because there's a huge difference with that one molecule. All right, anyway, we're still talking about monosaccharides. As a reminder, these are the monomers of polysaccharides, right? The monomers of carbohydrates. And they are the main fuels for cellular work. You do need to know that. Not even necessarily that that's going to be a test question, but you're going to be really familiar with that. So later in the semester, you're going to learn how our cells get energy to do work. And really the only one we're going to talk about for the most part is glucose. So when you learn about respiration and photosynthesis, it's going to be all about glucose. But in all actuality, you know, your cells do get energy from elsewhere. But that's the primary source of glucose. Not that this matters, so this won't be a test question, but just so you know, a lot of times when these monosaccharides get in water, they form rings. Again, I'm not going to test you on that, but if you're really paying attention, then I wanted to bring that up because I don't want you to get confused. For example, this is what glucose looks like, as you've seen before, but then when you put it in water, it looks like this. So I just don't want you to get confused and think to yourself, wait a minute. Earlier in the chapter, or earlier in the slideshow, he showed me this and said it was glucose. Now he's showing me this and telling me it's glucose. What's going on? So that's the only reason I'm showing you now. You don't need to differentiate between the two. I just don't want you to get confused when you see me showing it different ways. And of course, like I said earlier, these are monosaccharides, right? These monosaccharides are monomers, so we're bringing them together. So this is how we're bringing them together, not like this. Right? We're bringing them together like this ring is attached to this ring, is attached to that ring, so on and so forth. So they do turn into a ring, and that is how they're able to act as monosaccharides. So any questions about monosaccharides? Okay. Really quickly, we're going to talk about the next step up. We're still talking about carbs. we're still talking about sugars, right? So the one we just finished talking about is called monosaccharides. It's the one sugar. He guesses what the next thing might be if we just finished talking about monosaccharides. What is it? Yep, the other one, correct. What is the other one? Any guesses? We just got to talk about monosaccharides, and now we're going to talk about that one. Good. I don't know if it rhymed. I'm not sure if you said it correctly, but that's okay, because even if you said the other one, that's okay. Next thing we're going to talk about disaccharides, and I think I may have even heard you say bisaccharides, and that's okay too. I mean, because if you can remember bisaccharides, that will never be on an exam. So as long as you remember bisaccharides, you're like, oh yeah, it's di, not bi. But same thing, right? Same prefix. Anyway, disaccharides are double sugars. And as you might imagine, they consist of two monosaccharides. So you put one and one together, you get two. Nice basic math. Well, in biology, in this case, you put a mono and a mono together, you get a di. And here's some examples. Sucrose, which is made up of a glucose and a fructose. Lactose, which is made up of a galactose and a glucose. Maltose, which is two glucoses. You don't need to know any of those. Those are just examples. So here's a quick pop quiz to see if anybody can remember. Not that I expect you to yet, because I've only told you once, but we'll see if anybody can remember. If we have two monosaccharides, and we have to bring them together to build these disaccharides, what kind of chemical reaction is that when we bring them together? That's okay. Just wanted to see. Anyway, if you download the PowerPoint, you can watch this animation of it happening. And here's another hint. See if anybody can guess what kind of reaction this is now. And here's your hint. That's your hint. Any guesses? Dehydration reaction, right? Anytime we're building something, anytime we're bringing something together and taking water out of the reactants and into the products, that's a dehydration reaction. But anyway, yeah, that's just another example from the old textbook. Like I said, lactose is made up of glucose and galactose. There you go. So when you're drinking milk or eating ice cream or whatever dairy product you're working with here, it's a little sweet maybe, and that's where the sugar comes from lactose. I'm sure you guys have heard the term lactose intolerant, things like that. Here's some more examples that you don't necessarily need to know. But again, like we already said, you can find lactose in milk. You can find maltose in beer, and malted milkshakes, malted milk ball candy. I forgot this. I think they're called Whoppers. I don't remember. But anyway, and then the big one, and this is the one you might need to know if anything. If I do ask any examples of disaccharides, it's this one, because it's in our everyday life. So when people talk about sugar, usually they mean table sugar, right? So in this case, this is what we're talking about. Sucrose. It's a disaccharide. You need to know that. Questions so far? All right, I'm going to teach you a little bit more about disaccharides. I'm not going to test you on this, but it's good information for your everyday life. It was in your old textbook, so I kept it in here. Sucrose happens to be the main carbohydrate in plant sap. So, for example, real, what do you call that? Maple syrup, right? Real maple syrup is mostly sucrose. However, then you have something called high fructose corn syrup. I'm sure you've heard of it before. It's made by a commercial process that converts glucose into, like I said earlier, fructose, right? Fructose is sweeter, just because that one small difference in the molecule, right? That one small change in the shape. Fructose is a lot sweeter, so anybody who uses this can save a lot of money because this is a lot sweeter, therefore they can use a lot less of it. And because of that, it's often one of the first ingredients listed in soft drinks. For example, what is the first, well, it might be water. You got a Dr. Pepper. What's the first, what are the first three ingredients?
Carbonated water, high fructose corn syrup, caramel color.
Yeah, so it's the second one, right? It's mostly water. But then the next thing is not even flavors, right? Not even that, whatever gives it that Dr. Pepper flavor. The number 2 ingredient is fructose. It's interesting. I wish we had more time. You could look into it for extra credit. High fructose corn syrup is bad, but it's not nearly as bad as some people are making it out to be. Like I said, one of the few benefits of it is because it's so sweet, and it has the same molecular formula as glucose, but anyway, because it's so sweet, you can use less of it. So, obviously there's an advantage, not just to saving money, but also to your health. If you're using less sugar, that has some benefits. But anyway, here's what we're talking about. So why do they call it corn syrup? Also, there's a difference between high fructose corn syrup and corn syrup. I don't have time to talk about that right now. Anyway, so they take the corn, they extract the starch. Hopefully when you think of corn, you think of starch, as you should. And they break the starch down into what builds it, which I haven't taught you yet, and that's built of glucose, right? So we take that glucose, remember it only had that slight difference in change in the shape. They make that slight change, and that converts it to fructose, and that's what's being used now. Some more information. It's not in your textbooks, so I'm not going to test you on it. But again, for your everyday life, for your health and well-being, these are things you should know. The average American consumes about 130 pounds of sugar a year. Imagine that. Imagine like a 100-pound bag of sugar and how big that would be. That's how much the average American consumes. Because they put it in everything. You're like, oh, I'm eating healthy. All I'm having is, pasta sauce. And they put it in your pasta sauce, right? They put it in everything. It's a major cause of tooth decay and overconsumption. It also increases the risk of type 2 diabetes and heart disease. And basically, they're called empty calories because they do give you calories, which is what your cells need to work, but they don't give you any of the other nutrients that you need, which we'll also talk about later. So I didn't end there early at all. When we get back on Monday, we'll finish the discussion of carbohydrates by talking about polysaccharides, and then we'll move forward to the rest of them. That's it. You guys have a great weekend. I will be online.